to follow when it makes the complementary strand to these single-stranded DNAs here. One of the rules is that the enzyme has to make the new complementary strand always in the 5' to 3' direction. And this shouldn't be too shocking, because remember I always said that this is always the beginning. This is the 5' end of a piece of DNA is always where you start building a DNA polymer from, and the 3' end is always considered to be the finishing end of a strand of DNA. So what we're going to end up with is when this DNA polymerase III enzyme makes the new complementary strand up here, it's going to have to start off with the 5' end of that complementary strand and we'll build it like that, in that direction there. So the new strands are always made starting the 5' end and working towards the 3' end. But since double-stranded DNA is always going in opposite directions, the DNA polymerase III enzyme has to latch on to the 3' end of the template strand so that it can start the new complementary strand at the complementary strand's 5' end. To see if you got that, let's look at this other template strand down here. Is the DNA polymerase III going to latch onto this end or this end? The 3' end or the 5' end? It's going to have to latch onto this end, the 3' end of the template strand, because it needs to begin at the new strand at the 5' end. New DNA strands are always made at the start of the 5'. And so remember, you always have to have the 3' end and the 5' end opposite each other. So the new complementary strand will be made like this. All right, so the new strand is always made starting at the new strand's 5' end, working towards the new strand's 3' end from here to here. But that means it has to run along the template strand's 3' end to the 5' end, since double-stranded DNA always goes in opposite directions. See, I told you it gets a little complicated. We're only like 15 minutes into the lecture period yet. Okay, well, let's watch it in action. Oh yeah, so that's one rule, that the enzyme always has to make its complementary strands from the complementary strand 5' to 3' end. The other rule that DNA polymerase III enzyme has to follow is this. The enzyme, the DNA polymerase III, can only begin making complementary strands from a double-stranded region of DNA. In other words, it can't just latch onto the single strands and start making the complementary strands of them. There's got to be a double-stranded region where it begins. But there is none, because I just told you that the helix had separated, in the original double-stranded DNA separated into single strands. So there is no double-stranded region for it to begin. You know, if it was a racer, it needs a starting line to begin, and there is no starting line. There is no double-stranded. Well, what to do about that? Well, there's another enzyme involved in this process called primase. And primase enzyme is able to latch onto single-stranded DNA and make a short complementary RNA piece to that single-stranded DNA. And that serves as a double-stranded starting point for DNA polymerase III. So watch this in action here. Here's the primase enzyme. The primase enzyme goes up here and is going to make a short RNA primer, a complementary RNA to that piece of single-stranded DNA. There it is. So the red is RNA here. And even though it's a piece of RNA, this still counts as double-stranded nucleotides, a double-stranded region. And so that's good enough. Any double-stranded region is going to be a good starting point for DNA polymerase. And so now DNA polymerase has a starting point thanks to that RNA primer. It can now latch onto the template strand up there and start making the complementary DNA strand. Like that. Awesome. All right. Now, the problem is that, yeah, we've replicated some of this DNA here. In other words, we have a lot of this chromosome is now double-stranded DNA, but this part right here near the start is not. I mean, this zone right here is RNA, and it's not even attached to the that part right there, so something has to be done about that. Well, there's another enzyme. Let's not worry about the name of this other enzyme, but this other enzyme can come along and tear off the RNA primer and replace it with DNA. Here it comes. So now, at least this is double-stranded DNA here, where the RNA primer was. But the last problem is it's not linked up to the other newly made strand that DNA polymerase III just made. There's a gap in the backbone right there. Who remembers what molecules the backbone of DNA is made out of? Phosphate remember the backbone is phosphate deoxyribose, phosphate deoxyribose, phosphate deoxyribose, phosphate deoxyribose, that sort of thing. Well, there's a gap in the backbone there because that was the primer. And so lastly, there's yet another enzyme that comes along called ligase. And its name comes from a Latin word meaning to join together, because that's what this DNA ligase does. It can take two pieces of DNA and link their backbones together to create one piece of DNA. There we go. And ta-da, if you look at the top now, we've reached our goal. We now used to have one piece of single-stranded DNA, the dark blue one there, and now we have complementary double-stranded DNA. So we've replicated that chromatid, so to speak. But what happens next is, same thing takes place on this bottom template strand right here, and we're going to end up also with double-stranded DNA down there. So here we go. We need the starting point, the double-stranded region. Oh, well, first of all, let's bring in the next DNA polymerase III enzyme. Here comes primase to make a short double-stranded region of RNA. Now there's a double-stranded region, and the DNA polymerase III latches onto the template strand there and makes a new complementary DNA strand, 5' to 3' on the new strand. But now this RNA primer has to be torn off and replaced by a DNA primer, or I should say just by DNA. There we go. And lastly, the ligase enzyme has to come along and link the backbones together. There we go. And voila! We've now gone from an unduplicated chromosome to a duplicated chromosome with two identical sister chromatids. Thank you very much, DNA polymerase III enzymes. Your job is done. And here's the centromere back. Yes?
Why can't the DNA polymerase just start at the beginning and make it all by itself?
It sounds like you're essentially asking me, why is a primer necessary?
Yes.
Okay, yeah. I'm not sure. It's just not in one of the enzyme's capabilities to do that. Remember that enzymes, as amazing as they are, usually can just do one reaction. And so this particular enzyme, it's one thing it can do is to take individual nucleotides and add them on to a pre-existing double-stranded region. But yeah, it needs a primer of some sort, and I guess I don't know why exactly it needs a primer, but it does. Good question. Okay, a couple of points here. Notice that when a chromosome has, well, let me put this centromere back in. Where are you, centromere? There you are. So notice when a chromosome has duplicated, well, you know it has two identical sister chromatids attached to one centromere. It's considered one chromosome. You know that much. Notice that each new sister chromatid has one of the original DNA strands and one newly made DNA strand, the lighter blue one right there. And the same down here. Here's the original one in the darker blue that was present in the original chromosome, the unduplicated chromosome. And here's the newly made one in lighter blue right there. Since then in each new chromatid there's something from the old one that's been conserved, that strand, and something new, the newly made strand, they refer to this as semi-conservative replication. And there's kind of interesting historical thing related to that term. When they were first studying how chromosomes duplicated, they knew that you went from one piece of double-stranded DNA. Maybe I'll draw that over here. They knew that the cell started off with unduplicated chromosomes, one piece of double-stranded DNA, and it became two sister chromatids like that. But they didn't know whether the two original pieces went to one chromatid and the two newly made pieces went to another chromatid like that, or whether you got each chromatid having one old piece and one new piece like you see there. They just didn't know. And so when they worked out the process and they found out that each chromatid does have one old strand and one new strand, they said, okay, it's half conserving the old strand, so it's semi-conservative replication. And just to make sure you're doubly, or that you totally understand this, let me give you that review question again, or that thought question again. Get rid of those ones. So there's the two separated single strands. If I am the DNA polymerase III enzyme, and I want to make the complementary strand to this piece, do I begin here at the 3' end or here at the 5' end of the template strand? 3' end, right? Because the DNA polymerase III always has to latch onto the 3' end because it has to begin its complementary strand at the 5' end of the complementary strand. So it would go in this direction. And on this template strand, if I am DNA polymerase III, do I begin with the 3' end or the 5' end? I begin with the 3' end because the new complementary strand I'm going to be making has to begin at the complementary strand's 5' end, which always matches up to the 3' end of the original strand. So I would make my complementary strand in this direction there. So notice that the DNA polymerase III enzymes work in opposite directions. One starts on this upper template strand and moves to the left. One starts on this lower template strand and moves to the right. They're always traveling in opposite directions as they replicate the DNA. You almost think of at them waving at each other. How you doing? I'm doing fine. How you doing? As they pass each other up, making the new strands. And so that little fact that when the DNA is being replicated, when the chromosomes are being duplicated, the two DNA polymerase enzymes move in opposite directions is going to become important here in a minute. So I just want to verify that you know it. Okay, now this thing that I've been showing you here with the absurdly short chromosome is very much just sort of an introduction to this topic of how the chromosomes duplicate themselves. But it does cover some of the major points, that the DNA polymerase III is the main enzyme to do it, but it takes other enzymes as the primase enzyme to make the RNA primer. There's another enzyme that rips off the primer and makes changes in the DNA, and another one that ligates the two pieces of the primer, well, what used to be the primer, but is now DNA, to the complementary DNA strand. It's really whole teams of enzymes that are involved in this process. And when I've shown you this diagram here, you would almost get the idea that one DNA polymerase III enzyme started at the extreme right end of the chromosome and worked its way all the way over to the extreme left-hand end of the chromosome on that strand, and then another DNA polymerase III enzyme started at the extreme left end of this chromatid and worked its way all the way across the chromosome to the extreme right-hand end. That was a simplification. The fact of the matter is that chromosomes are so long, it would just take too long for any one DNA polymerase III enzyme to work its way all the way across. Your cells would take days and days and days to replicate themselves. And so the fact is that there's more than one spot along the chromosome where this process is occurring. To make an analogy, if there was a super long stretch of highway, like Highway 5 or something like that, and the whole thing needed to have an extra lane added to it, they would probably have several work crews all along Highway 5, each adding the extra lane at various spots. And so that's more of the way that the chromosomes actually duplicate, and that's what I'm showing you here. This is supposed to be a chromosome now. There's the centromere, and these are the dark blue is the original chromatid, the double-stranded DNA there. When the chromosome is going to be duplicated, several spots along that original chromatid called origins of replication open up, and at each of those origins of replication, you're going to have, like this, you're going to have teams of DNA polymerase 3 and primase and ligase and all the enzymes that are involved in that I just showed you. You have teams of them working here, and then another teams of them working here, and more teams of them working here. And even this is something of a simplification. Here on this chromosome, I'm only showing you 3 origins of replication, but in real chromosomes, you might find hundreds of these origins of replication all throughout the original chromatid, and in each one of those spots, the DNA is being duplicated. All right, and these